Hello everybody, today we're going to talk about enzymes and inhibition. Inside every living cells, we have enzymes that act like workers, speeding up chemical reactions and keeping everything running smoothly. But sometimes, other molecules can interrupt this process and slow it down, even stop it from working altogether. This process is called enzyme inhibition. In this video, we'll explore the two main types of inhibition, competitive and non-competitive inhibition. But before we begin, let's quickly go over what an enzyme is. An enzyme is a protein that speeds up chemical reactions in our cells. Each enzyme has its unique shape, and the most crucial part is the active site. At the active site, the substrate, which is the molecule the enzyme acts on, fits perfectly and helps the enzyme convert it into a product. Once the product is formed, it is ready to work again. Now let's talk about normal inhibition. Imagine a reserved parking spot, which is the enzyme's active site. The spot is waiting for the right car. The substrate, the vehicle, fits perfectly, pulls in, parks, and the enzyme helps transform it into a product. When the product is released, the parking spot becomes free again for the next car. In our case, it would be the next substrate molecule. However, sometimes we have people who are in a hurry and park whenever they see a free spot, even if it isn't intended for them. The same thing happens in our body. Some molecules called inhibitors interfere with the normal process of enzyme activity. In competitive inhibition, two molecules, the substrate and the inhibitor, compete for the same site on the enzyme, known as the active site. Now imagine the enzyme's active site is reserved parking space. The substrate is the car that is supposed to park there, but along comes another car, which is the inhibitor. It looks very similar to the original one and steals the parking spot. So if the another car parks there, the real car can't get in. It means no product or less product is made. One real-life example is caffeine. In the brain, we have a molecule called adenosine, which normally binds to its receptor to make you feel tired. However, caffeine resembles adenosine closely, so they both compete for the same binding spot. When caffeine takes the parking spot, adenosine can't bind, which is why you feel more awake after drinking coffee. On the other hand, non-competitive inhibition is slightly different. In this case, the cars don't compete with each other for the same reserved parking spot. Instead, there is a big truck parked on the side, blocking the driveway of the reserved parking spot. Even though the parking spot is open, the car can't get in as the truck is blocking the driveway. This is similar to what happens in the body when an inhibitor binds to a different site on the enzyme, changing the enzyme's shape and blocking access. As a result, the substrate cannot perform its function and no product to less product is formed. One real-life example of non-competitive inhibition is when fluoride affects the enzyme enolase in our body. Enolase is important in glycolysis, which is the process that breaks down glucose to produce energy. When fluoride binds to enolase, not at the active site, it changes the enzyme's shape, preventing it from helping in glycolysis. Now, let's do a quick recap of everything we've learned today. In competitive inhibition, the inhibitor competes with the substrate for the same parking spot. If the wrong car parks first, the real car cannot enter and no product to less product is formed. The inhibitor blocks the active site, preventing the substrate from binding. Now, to non-competitive inhibition, the inhibitor blocks the parking spot from the side. Even though the parking spot is open, the substrate can't get in. The inhibitor binds to a different site on the enzyme, which alters the shape of the active site, which means the substrate can't bind properly and no product to less product is produced. Both types of inhibition help us regulate enzyme activities in our cell, which is really important. However, understanding this mechanism is also essential for designing drugs and controlling biological processes. That's it for today. Thank you for watching. For more content, please go to my YouTube channel.